Welcome to the Dye-Bespattered Genius, an exploration into the life, character, and colorful science of Paul Ehrlich. In this presentation, we journey into the world of a visionary pioneer who turned vibrant dyes and bold chemical concepts into breakthrough medical discoveries, forever changing the fields of hematology, immunology, and chemotherapy. In 1876, a professor dismissively remarked to Robert Koch that young Ehrlich was good at staining, but unlikely to pass his exams. Fast forward to 1908, and that same, little Ehrlich, was awarded the Nobel Prize in physiology or medicine. What started as an obsessive fascination with laboratory dyes evolved into a medical revolution, earning him lasting honor as the father of targeted chemotherapy. It is a striking journey from early academic skepticism to world-changing scientific triumph. Born in 1854 in Strehlen, Silesia, Paul Ehrlich combined his father's excitable energy with his mother's strict organizational discipline. Already concocting homemade cough drops by age 8, Ehrlich found his true scientific spark at 14 through his older cousin, pathologist Carl Weigert. Weigert introduced the young prodigy to the microtome and the vibrant world of synthetic aniline dyes, launching a passion that would ultimately transform modern medicine. Paul Ehrlich was hardly a model medical student. Refusing to memorize over 10,000 required terms and skipping clinical rounds, he spent his days buried in laboratory experiments, leaving his hands and clothes permanently stained with dyes. Yet this singular rebellion paid off. In 1878, at just 24 years old, Ehrlich earned his medical degree from Leipzig. Not through conventional rote learning, but on the strength of a groundbreaking thesis exploring the chemical affinities of biological stains. Ehrlich famously described himself as a monoman, a figure driven by a single, uncompromising devotion to scientific advancement. Fueled by 25 strong cigars daily and mineral water, he routinely forgot to eat. Any surface became his canvas. He frantically scribbled molecular formulas on lab walls, menus, tablecloths, and even the sole of his shoe while traveling. Though socially shy and absent-minded, he became a force of nature at scientific meetings, launching into furious, cigar-waving debates whenever his passion took hold. Behind what seemed like a chaotic clutter of test tubes and stacked manuscripts lay Paul Ehrlich's perfectly tuned machine. He governed his institute through a strict color-coded system, scrawling non-negotiable daily instructions in colored pencil onto paper blocks, a distinct hue for each department. Demanding immediate action and daily progress reports, this ruthless method left no room for hesitation. Only those willing to execute Ehrlich's extraordinary, intuitive leaps could survive in his relentless pursuit of scientific progress. Behind every eccentric genius stands a steadfast anchor. When Paul Ehrlich met 18-year-old Hedwig Pincus, he famously arrived carrying 3 top hats and sporting dye-stained fingers. Despite an unpromising first impression, their marriage proved to be his ultimate shield. Hedwig managed every domestic detail, tolerated his quirks, and even funded his private laboratory research. By shielding him from everyday worries, she provided the essential foundation that allowed Ehrlich's brilliant scientific mind to thrive. At the heart of Ehrlich's work was one central question: why do different biological tissues have distinct affinities for the same dye? This thread of color guided his career through 3 monumental phases. It began with staining dead cells, giving rise to clinical hematology. It evolved into studying interactions with living cells, unlocking cellular immunology. Ultimately, it led to targeting and destroying pathogens, founding modern chemotherapy. A single, unifying obsession transformed 3 separate fields of medicine. By systematically classifying synthetic dyes into basic, acidic, and neutral categories, Ehrlich unlocked a hidden cellular world. His precise techniques allowed him to discover and name the mast cell and eosinophil, making him the first to differentiate various white blood cells. This colorful taxonomy laid the foundation for modern clinical hematology, enabling the very first true diagnostics for leukemias and anemias. Shifting from dyes to immunity, Ehrlich proposed that biological interactions are fundamentally chemical. He theorized that cells feature specialized receptors, or side chains, designed to absorb nutrients. When invading toxins hijack these receptors, the cell overcompensates by producing a flood of excess side chains. Released into the bloodstream as antibodies, they neutralize the threat. This groundbreaking model of cellular immunity earned Ehrlich the 1908 Nobel Prize. By the late 19th century, Louis Pasteur and Robert Koch had proven that microbes cause infectious disease. Yet a monumental paradox remained: when invaded by deadly pathogens, how does the host manage to survive? In search of an answer, the scientific world fractured into two opposing camps, setting off a classic battle of biology versus chemistry to unlock the secrets of immunity. In 1882 in Messina, Italy, Russian zoologist Elie Mechnikov made a breakthrough while examining transparent starfish larvae under his microscope. Piercing a larva with a tiny splinter, he watched as mobile cells rushed to the site of injury. Rather than spreading disease, as was widely believed, these eating cells were actively defending the host. Framing this response as physiological inflammation, Mechnikov revealed a living, cellular defense system. An internal Darwinian struggle that established the biological side of immunology. Mechnikov named these cellular defenders phagocytes, literally eating cells, dividing them into large macrophages and smaller microphages, known today as neutrophils. Through a precise three-step sequence of recognition, engulfment, and digestion, these cells seek out invading pathogens and destroy them from within. By detailing this dynamic process of phagocytosis, Mechnikov established the bedrock of innate cellular immunity, revealing how the body actively hunts and consumes microscopic threats. While Mechnikov saw biology, Paul Ehrlich saw structural chemistry. Building on his 1878 thesis on histological staining, Ehrlich observed how synthetic dyes selectively color specific tissues, such as methylene blue targeting nerve cells. From this, he realized that chemical interactions govern all biological life. Unbothered by elaborate setups, Ehrlich declared he needed only a water tap, a flame, and blotting paper to work, even in a barn, driven by the firm belief that chemistry holds the ultimate key to understanding living systems. Working alongside Emil von Behring at the Koch Institute, Ehrlich investigated how animals survived diphtheria. They discovered that immunity was driven not by living cells, but by soluble chemical antitoxins circulating within the blood serum. Bringing his signature quantitative rigor to this breakthrough, Ehrlich established exact standardizations for serum potency. By transforming raw serum into a reliably measured medical treatment, he laid the foundation for humoral immunology and made widespread, life-saving serum therapy possible. In 1897, Paul Ehrlich introduced his side chain theory to explain how invisible chemicals neutralize specific toxins. He proposed that cells possess macromolecular side chains meant for binding essential nutrients. Toxins, acting as chemical mimics, use an anchoring group called a haptophore to hijack these receptors through precise chemical affinity. Operating like a lock and key, this landmark concept revealed how molecular interactions drive cellular immunity. When a toxin permanently docks into a cell's side chain, it blocks normal metabolic uptake. To overcome this blockage, the cell overcompensates through hyperregeneration, producing a massive surplus of identical receptors. Unable to hold this overwhelming abundance, the cell sheds the excess side chains into the bloodstream as superfluous ballast. Once free-floating in circulation, these detached receptors become circulating antibodies, capable of seeking out and neutralizing toxins before they ever reach the cell. In 1901, Ehrlich confronted a disturbing dilemma arising from his own theories. If the body manufactures precise molecular weapons, what stops it from turning those weapons against itself? He termed this threat horror autotoxicus, the fear of self-poisoning. Ehrlich postulated that healthy organisms must maintain strict internal safeguards to prevent self-destruction, a concept that laid the theoretical groundwork for modern research into immune tolerance and autoimmune disease. For over a decade, a bitter scientific rivalry divided Europe. In Paris at the Pasteur Institute, Elie Mechnikov championed biology, arguing that mobile cells called phagocytes drove innate immunity. Meanwhile, in Berlin at the Koch Institute, Paul Ehrlich countered with chemistry, contending that circulating antibodies and side chains in blood serum governed adaptive immunity. Each school believed its mechanism was the singular driver of host defense, setting off a monumental debate between cellular biology and chemical affinity that defined the dawn of modern immunology. In 1908, the Nobel Committee awarded the prize in physiology or medicine jointly to Mechnikov and Ehrlich, recognizing that immunity is not an either/or proposition. True defense lies in synthesis. Ehrlich's circulating antibodies coat and tag invading pathogens through opsonization, which dramatically accelerates the ability of Mechnikov's phagocytes to recognize and devour the threat. Ultimately, chemistry and cellular biology proved to be two essential sides of the exact same life-saving coin. Ehrlich realized that if side chains could target toxins, synthetic chemicals could be engineered to destroy microbes without harming the host, a concept he termed the Zauberkugel, or magic bullet. In 1909, collaborating with Soichiro Hata to screen hundreds of arsenic compounds, Ehrlich discovered compound 606, known as Salvarsan. Delivering the first effective cure for syphilis, this breakthrough turned his vision into reality and marked the official birth of targeted chemotherapy. Driven by a bold vision, Paul Ehrlich launched his quest for the magic bullet, a concept he called therapia sterilisans magna, or complete sterilization in a single blow. He made an audacious leap of logic. If synthetic dyes could selectively stain specific tissues while ignoring others, then targeted chemical compounds could be engineered to seek out and destroy invading pathogens while leaving the human host entirely unharmed. This fundamental question reshaped medicine forever. Collaborating with Japanese bacteriologist Soichiro Hata, Paul Ehrlich launched history's first systematic, high-throughput drug screening. Testing hundreds of toxic arsenic compounds against syphilis, success arrived on their 606th attempt with arsphenamine, marketed as Salvarsan. Ehrlich attributed this historic triumph to his famous formula of the 4Gs, Geld, Geduld, Geschick, and Glück, money, patience, skill, and luck. This relentless pursuit delivered a revolutionary cure and forever changed pharmaceutical research. For centuries, humanity stood defenseless against Treponema pallidum, the spiral bacterium responsible for syphilis. This devastating pathogen inflicted widespread suffering across generations, bringing dementia, paralysis, and death. Yet, the prevailing standard of care offered little solace. To combat the infection, patients endured the grueling mercury standard, a brutal 2 to 4 year regimen of toxic heavy metal injections. Efficacy remained tragically low while toxicity was severe, leaving medicine locked in a desperate struggle against a relentless scourge. Working with aniline coal tar dyes, Ehrlich observed that specific chemicals selectively stained distinct cells without marking surrounding tissues. This inspired a bold leap: if a chemical could stain a specific microbe, could one be engineered to selectively destroy it? Early triumphs soon proved the concept when methylene blue targeted malaria in 1891 and trypan red cured sleeping sickness in mice in 1904. By shifting from staining cells to hunting pathogens, the era of targeted chemical affinity was born. This vision birthed the Zauberkugel, or magic bullet, principle, a drug designed to strike disease with absolute precision. To succeed, a compound required two critical properties: it had to be strongly parasitotropic, possessing maximum killing power against invading pathogens, while remaining minimally organotropic to spare host cells. The underlying mechanism relied on specific chemoreceptors on the pathogen. By tailoring drugs with specialized molecular appendages, these magic bullets could anchor exclusively to microbial receptors, eliminating the target while leaving the body's healthy tissues completely unharmed. Paul Ehrlich and chief chemist Alfred Bertheim turned their focus to atoxyl, an arsenic compound that killed trypanosomes but tragically caused blindness. Proving its chemical structure could be modified, they launched a massive, systematic iteration engine. Hundreds of variants were synthesized and tested in relentless sequence. By compound 418, arsenophenylglycine, they unlocked an effective treatment for sleeping sickness. Yet, their ambitious hunt for a syphilis cure demanded even greater perseverance, pushing their systematic testing far into the 600s. In 1909, a crucial missing link arrived in Frankfurt from Tokyo: Dr. Soichiro Hata, a pupil of renowned bacteriologist Shibasaburo Kitasato. Hata brought a game-changing breakthrough, a reliable method for experimentally infecting rabbits with syphilis. With this vital animal model finally established, Ehrlich assigned Hata to painstakingly retest every single compound they had previously synthesized against the disease, setting the stage for a dramatic breakthrough. First synthesized in 1907, compound 606, arsphenamine, was initially shelved after an assistant erroneously reported it had no effect on trypanosomes. However, on August 31, 1909, Dr. Hata's meticulous retesting brought a monumental revelation: 606 possessed an incredible therapeutic index for syphilis. The results were astounding: a single dose achieved complete cures in syphilitic rabbits within just 3 weeks, with 0 animal fatalities. The once discarded chemical was officially resurrected as a miraculous medical triumph. On April 19, 1910, an electrifying announcement in Wiesbaden stunned the medical community. Dr. Schreiber of Magdeburg Hospital confirmed the first successful human treatments using compound 606. The clinical results were miraculous. Terminal patients suffering from dementia showed dramatic improvement, while early-stage patients watched their infected sores completely vanish. Following this extraordinary validation, compound 606 was officially registered under the trademark Salvarsan, the arsenic that saves, marking a monumental turning point in modern medicine. Salvarsan marked a monumental paradigm shift in pharmacology. Where the empirical mercury standard meant severe heavy metal poisoning and 2 to 4 years of unpredictable, suppressive treatment, compound 606 was rationally designed around receptor theory. With low host toxicity and rapid targeted action, Salvarsan slashed treatment duration to just weeks or months. It transformed clinical outcomes from mere suppression to a true cure, achieving therapia sterilisans magna and permanently redefining medicine. Demand for the new cure erupted instantly, far outstripping early laboratory capabilities. To ensure rigorous testing and collect crucial clinical data, Ehrlich provided 65,000 doses to physicians worldwide completely free of charge within just 1 year. Backed by the George Speyer House and Hoechst Chemical Works, production expanded on an extraordinary global scale. By 1923, 2 million doses per year were being manufactured in the United States alone, delivering medicine's first magic bullet worldwide. Despite its breakthrough success, Salvarsan faced fierce resistance during the Salvarsan War. Because intravenous administration was complex, physicians who ignored Ehrlich's precise instructions accidentally caused tissue necrosis and patient deaths. Public backlash erupted as sensationalist media, including the newspaper Die Wahrheit, viciously accused Ehrlich of fraud, charlatanism, and unethical testing on prostitutes. Although a 1914 libel trial fully exonerated Salvarsan and jailed the publisher, the relentless, anti-Semitic attacks took a tragic toll, permanently breaking Ehrlich's health. To overcome Salvarsan's complex preparation, acidity, and oxidizability, Ehrlich returned to the laboratory, synthesizing through the 700s and 800s to solve its usability challenges. His continuous iteration yielded compound 914, introduced in 1912 as Neo-Salvarsan. Though slightly less active against the spirochete, Neo-Salvarsan was water-soluble, infinitely safer, and vastly easier for average physicians to administer. This refined breakthrough successfully solved the administration crisis and remained the global standard for 30 years. Salvarsan served as the definitive proof of concept, establishing a system of synthesis, animal screening, evaluation, and modification. Ehrlich didn't just invent a drug, he created the pharmaceutical screening framework itself. This breakthrough directly inspired Gerhard Domach's discovery of sulfa drugs and spurred Alexander Fleming's hunt for penicillin. Today, from monoclonal antibodies to targeted immunotherapies, all modern targeted treatments trace their conceptual lineage back to the Zauberkugel. As an architect of modern medicine, Paul Ehrlich transformed 3 distinct fields through targeted mechanisms. In hematology, selective dye affinity for blood cells revolutionized leukemia and anemia diagnostics. In immunology, receptor and side chain binding to toxins and antigens led to antibody and vaccine standardization. In chemotherapy, targeted chemical toxicity against spirochetes and pathogens laid the groundwork for modern antimicrobials and oncology. Together, these breakthroughs established an enduring legacy, bridging diagnosis, immunity, and targeted therapy. Despite global fame, Paul Ehrlich remained a tireless monoman, but his relentless obsession exacted a heavy toll. Years of intense research, the stress of defending Salvarsan in court, and smoking 25 cigars a day severely compromised his cardiovascular health. Forced by doctors to abandon both work and smoking, he fell into deep depression, suffering profoundly from constraint. Following 2 strokes, Ehrlich passed away in August 1915 at age 61, surrounded by his protective family, leaving behind an extraordinary scientific legacy forged through absolute devotion. Paul Ehrlich summarized his philosophy through the 4 G's of scientific achievement: Geld, Geduld, Geschick, and Glück, money, patience, skill, and luck. A towering genius who revolutionized histology, immunology, and pharmacology, Ehrlich opened doors to the unknown. Ultimately, he proved that medicine could confront and conquer pathogens on their own ground at the molecular level, leaving an enduring legacy that permanently transformed modern medicine. From a shy boy playing with colorful dyes in a Silesian tavern, Paul Ehrlich rose to become the father of targeted medicine. His early passion for chemical stains paved the way for today's advanced molecular therapies. Every time a physician orders a blood smear, administers an antiserum, or prescribes a targeted drug therapy, they are following the vibrant blueprint drawn by Paul Ehrlich, an immortal legacy that forever transformed the science of healing.